Hello, my name is Mike Shane, and I'm a research scientist for the Hubbs-SeaWorld Research Institute. I'm also the Director of Replenishment Operations and the Diving Safety Officer for our organization. I've been with Hubbs now, or at least working under this programs, for 30 years now, and I did my undergraduate work at San Diego State as well as my graduate work at San Diego State University in marine ecology.
Where are we now, Mike?
So right now we're at our marine fish hatchery in Carlsbad, California. It's the only marine fish hatchery on the west coast of the United States. And what we're looking at right here is a machine and how we tag our cultured fish. Fish are cultured here at the hatchery, and before they leave the facility, a tag is inserted to each fish to help us identify them once they're recovered in the future. We'll talk about that a little bit later. But this machine here is the tagging machine that inserts the tag into a 1 millimeter small piece of wire, smaller than a grain of rice, into each individual fish. So a fish that, this is the machine here, there's a spool of wire here, it travels through the machine, and actually this needle on the end, the fish is, head of the fish is in, this needle is put on into their cheek muscle, tag is injected into the fish, and then we check it for a tag. So the tag actually looks like, and here's a model of a fish. This is actually a salmon, but inside that yellow circle, oval there, is actually the actual tag that's inserted into each fish. If you look at that tag under a microscope, you can actually see there's numbers etched on it. Here's a blown up version of it, a little hard to see, but there are actually numbers etched on it. Each individual fish or group of fish has a unique set of numbers on it that identify that fish coming from the hatchery and its whole process and where the fish came from. And we ask fishermen, once these fish recruit to the fishery, that means once they reach 28 inches in length, they can be caught by recreational and commercial fishermen and to feed their families, friends, etc. This is actually something that we put together years ago to remind them to help cut the heads off and save it for us. So the tag sits up here behind the eye, and the cheek muscle is where the tag is. So once they cut the head off, they drop them off at freezers up and down the coastline. We can then use this device here to then pass it over the head of the fish and check if it's tagged. And this beeps, and then we have to dissect that little tag out of the cheek muscle of those fish.
What is the information on the tag? Would you kind of go specific? What is it?
So the information on the tag, really we have to go back to our database. It's just a unique number. For example, 78. So we go back into our database. Okay, what's 78? What does that mean? And we go back and say, hey, this means it came from this crop of fish. We released it X number of years ago. For example, we just got a fish back a year ago that had been out there for 20 years running around the ocean. Pretty significant. So we can go back from 20 years ago and look and see the history of that fish. Which broodstock tank did it come from? Which tanks did it grow up in? And what grow-out facility did it go to prior to being released? And we'll talk a little bit more about the grow-out facilities once we go out and look at our grow-out pen outside here.
When these fish are released, how far will they travel?
This species of fish is considered a coastal pelagic fish. They can travel miles. I mean, we've seen fish released from the hatchery that are, again, small little 8-inch fish, are basically 20-30 miles up the coastline in a matter of days, even as youngsters. So the adults, we know, probably they roam quite a bit. I mean, they roam throughout Southern California, up to Monterey, even Bodega Bay. They've been seen as far north, at least wild fish have, as far as southeast Alaska. But typically the range is up to Northern California, down into Baja. And we know that these fish move around quite a bit because the genetics indicates that it's one big population out there. There are no sort of subpopulations of sea bass. To be able to be considered panmictic, or one big population, these fish have to move around a lot, and a lot of exchanging of genes going on out there. So, you know, these fish go up and down a broad range of thousands of kilometers along the coast here of North America.
So describe what it was. You mentioned a subspecies or a
subpopulation. So, for example, what I mean is the sea bass genetically, a sea bass caught off of Monterey versus a sea bass caught off of San Diego versus a sea bass caught in Mexico. There's no difference genetically among those fish. A subpopulation would be, okay, we could genetically distinguish a fish that's caught off of, say, Northern California versus a fish that's caught off of San Diego. Because this species moves around a lot, we do not see that sort of subpopulation like we suspect in other, and we know of in other species of fish. For example, the California halibut is a species that we think there's some subpopulations or a little bit of different genetic structure to fish in different parts of the coastline where they occur.
Because they don't travel as far.
Because they do not travel as far, correct.
All right, so now we're going to go out into the tagging area, right?
Yes, we'll take a look at how we use these machines, and they're actually, when we're tagging, in the event of tagging, we have multiple machines, as you can see, and there's a specific room that we tag in the hatchery when it's time to tag our fish.
Let's go there now. Where are we now, Mike?
So we're in the tagging room. This is where we tag the thousands of fish, millions of fish that we've released over the 30-year history of this program. So this is where it all starts. So we bring fish from our tanks in the other room, and we sluice them through pipes gently into this holding tank. There's hundreds of fish that are swimming around in this holding tank. As it comes time to tag, we then move fish a small netful at a time and set them into these two different troughs in here.
How big are they, Mike, when they're going through this process?
Roughly about 3 to 4 inches in size.
How old?
And about 90, 100 days old, roughly on average.
Okay.
3 to 4 months when we're tagging them. So once the fish come out of a few dozen at a time, will be set into here. And this system has recirculating water that has anesthesia in it, anesthetic. So it puts the fish to sleep to make it less stressful on the fish for the tagging that they're about to go through, in addition to making it easier for the taggers to handle the fish because they're not flopping around. So once the fish spend a few minutes in here, they go to sleep. We can then lift up the gate and allow them to flow down the race track here, and actually the taggers that are sitting at these stations here can then open their gate and allow the fish to come down to their holding tank right here. So now the tagger is sitting right here. The machines that we looked at earlier would be sitting right here with the needle right here. So the tagger would grab the fish out gently, orient them, make sure that needle, which is really fine gauge needle, I mean it's a small shot, a needle that you would kind of get that you may not even know, the doctor, so these fish don't even feel that. So the needle is put into their cheek muscle, they're tagged, and then the tagger drops them down this bin chute that has water flowing in it. As the tag passes down, or the tagged fish passes down this chute, this does the same thing as that blue wand did. It basically detects the tag. Down here, there's water jets that are going one direction or the other. As the fish passes down here, it detects the tag, changes the water jet, and shoots the fish in this direction. If for some reason the fish didn't get tagged, it actually travels to the left side here. It goes here, and we have to re-tag it or check it out and make sure what's going on. But once this fish passes down here, it enters a larger 3-inch piece of pipe, exits the building, and flows under the driveway out to tanks outside called raceways, where they're then held for several weeks prior to them going to the various grow-out pens.
How many people will be tagging during one project?
So we have the ability to have multiple, at least 3 or 4 stations here. So right now if you saw 3 machines, we have 3 stations set up. We were actually tagging last week and the week before, given the social distancing that we're all practicing right now. So we weren't using this station. We were just using this station and the far station, and we were practicing our social distancing because these are more than 6 feet apart. We had somebody over there, so we were able to still work through this room and keep everyone safe and healthy.
So how many fish how many fish can someone tag in a minute? I mean, obviously it's not a race, but what is it like?
Yeah, our tagging rate is anywhere 400, 500 to 600 fish an hour is basically kind of what we're tagging. So if you break that down in numbers, you can tag about 150 fish, every 15 minutes or so, so 300 every half hour. You can kind of look at your watch and kind of see your rate, but that's usually about the rate that you're doing, anywhere between 100 to 150 fish every 15 minutes.
So over the years that we've been doing this, how many fish have passed through this room?
Well, millions of fish. I mean, we released over 2.5 million fish. So this facility's been here for 20 of the 30 years that we've operated this program. So, you know, most of our fish have come through this facility. And this, again, this was what's unique about this and our program is this, you know, technique that we're tagging is a way for us to be responsible as scientists to understand what's going on with the fish after we release them. By tagging them, we're able to make assessments of survival rates and traveling. If we weren't doing that, we just wouldn't know where our fish are going if they're surviving. What you see here, this is a custom-built system. This is something I just can't flip through a catalog and put together. We built this with custom engineers and assembled with multiple meetings and design plans to put this system together. So this is all purpose-built specifically for our fish and this program.
Hey, where do they go from here?
So when they leave the tagging room, they then travel the pipe outside, goes outside this building, under the ground and outside to raceways, where these are outdoor tanks that have flow-through seawater, where we then hold them for about another couple, 2-3 weeks before they're then delivered to grow-out pens.
How about if we take a look?
Yeah, let's go take a look at one.
Where are we now, Mike?
So right now we are where the fish are come from the tagging room. Remember they traveled under the driveway, they come out this pipe, and then we can direct the pipe with the longer pieces that we put onto this to any one of the different raceways out here. And you can see all the fish that are in these raceways are fish that we've tagged over the last couple of weeks. So in this raceway, there are approximately 12,000 fish right now. There's about 6,000 in this half and about 6,000 in this half.
I understand the recent rains have made the water a little turbid, they say.
Yes, I mean, we had a historic week last week, and so fresh water and salt water obviously don't mix, and so with all the runoff in this coastal lagoon that happens from all the development up coast, up the hillside here, this is coming in, this is just runoff that, fortunately, the water's getting better now, but a couple of days ago it was even browner. So it's clearing up now. Fish are doing well. They can handle the reduced salinities that we were seeing here and still be fine. So now the salinities are coming back up. The water will start clearing up now that we're past beyond the rain event.
Okay. Where are we going now?
We're going to walk out and look at the grow-out pen that's outside here.
So this is the final stop after the tagging, after the raceway. How long are they in the raceway after the tagging?
Usually in the raceways for a couple, I mean, ideally we do it for a couple of weeks to assess the tag retention. We want to make sure the tags are staying in there, they're looking good, before we then deliver them to a grow-out site. And one of these, when there's 11 grow-out sites up and down the coastline, we operate 3 of those. The other ones are run by the Coastal Conservation Association, or CCA. Their volunteers have been actively engaged in this program, contributing thousands of hours to take care of fish in their systems, grow them to a larger size, and release them. So they're awesome stewards of the environment, helping to help this program be successful and get fish to a larger size before they're released.
Big ups to CCA, right?
Yes, big ups to CCA. I mean, they're huge, and there's no other program like this really in the United States where you have such a large input from volunteers helping a stock replenishment program. Usually those programs are run just by their own staff, and then the other organizations like CCA may help support it by catching fish, but they're actually involved in helping us grow fish out.
So what's on the other side of that fence?
So that's one of the grow-out sites. Let's take a look at it. So what you're looking at right here is a grow-out pen. There's actually 2 pens that we can hold anywhere, from 10 to 15,000 fish in each one of those grow-out pens until they get to be about 8 to 10 inches, and then we let them go. So just last week, we actually released fish right before the rain events came from both of those pens. We released about 12,000 fish out of those 2 pens. So these are pens that we, Hubbs-SeaWorld Research, operates and manages. We have 2 other pens, one in Catalina and one down in San Diego Bay that we also operate, but the other 6 sites are actually 8 sites, are run by the CCA volunteers. and help to put in thousands of fish themselves every year with growing out. For example, Channel Islands Harbor is one of the sites, and they're getting ready to release about 10,000 fish probably this week as well.
Hey, Mike, thanks.
You're welcome. Thank you.